Hi, my name is Alyson Hall, and I'm a PhD student at the Virginia Institute of Marine Science. I study seagrasses and the ecology of seagrass meadows, particularly in the Chesapeake Bay. My lesson plan is aimed at 7th grade life science students, but includes an extension activity that makes it applicable to high school biology students as well. The lesson focuses on watching how seagrasses breathe or measuring their rates of primary production and respiration. Students will learn what seagrasses are, how they differ from seaweeds, and what species of seagrass we have in the Chesapeake Bay. They'll then learn why seagrasses are important and why scientists care about measuring things like seagrass metabolism or the comparison and study of their rates of primary production and respiration. When I'm taking these measurements underwater in a seagrass meadow, I use these benthic incubation chambers to trap water around the grass. I use these black chambers that block out light to measure respiration, and I use these clear chambers that allow in light to measure net primary production. And I do that by using a dissolved oxygen sensor to track how the amount of dissolved oxygen changes over time. In this lesson, your students become the dissolved oxygen sensor and count bubbles of oxygen either leaving or entering the plants in each of these chambers. I provide different ways for students to count the bubbles, either including the provided videos or as a printable handout. Through this lesson, your students will learn to write hypotheses, measure and calculate rates of respiration, net primary production, and gross primary production, and then plot their results as a bar graph to compare the rates between our two seagrass species. The base lesson ends with students scaling up their rates from 1 hour, the amount of time our chambers run for, to rates for an entire day by multiplying by the number of hours in a day. The worksheet is split into 3 sections, and all throughout the students are going to be forming more hypotheses and answering discussion questions that relate back to seagrass ecology and seagrass meadows, and why which species is dominant in a meadow can really affect the entire ecosystem overall. There are slides that define terms that might be unfamiliar, like net autotrophic and net heterotrophic, that the students will use in their discussion questions. If you want to push your students, or you have a high school class, you can add the second worksheet as an extension to this lesson. The extension activity allows the students to think a little bit more like scientists and take their rates from one small chamber to rates for the entire lower Chesapeake Bay. They'll then think about how a system like the Chesapeake Bay might change over time in terms of the amount of grass that's there, and what that could mean for their rates of primary production and for respiration. In particular, students will understand that the amount of seagrass in the Chesapeake Bay is not constant year to year, and neither is the amount of each species. They will use data from the SAV monitoring lab here at the Virginia Institute of Marine Science to calculate rates of gross primary production in 1990 and in 2020 to compare these 2 years and then answer some more discussion questions. I hope this lesson leaves you and your students being just as excited about watching seagrasses breathe as I am. If you have any questions or want more information, please don't hesitate to reach out to me. Thank you so much for watching.